When a sequence is generated from ends of a single clone, the distance between the two opposing fragments of a clone is fixed to, meaning that they are always separated by a distance defined by a length, normally 1,000 to 9,000 bases. A. Both an uncertain range clone. B. One an uncertain range clone. C. Both a certain range clone. D. Both a certain range gene. The correct answer is both a certain range clone. Which of the following is incorrect about ARachNE? A. It accepts base calls with associated quality scores assigned by Fred as input. B. It is a free UNIX program. C. It is for the assembly of whole genome shotgun reads. D. It doesn't involve heuristic approach. The correct answer is it doesn't involve heuristic approach. Which of the following is incorrect about FRAP? A. It aligns individual fragments in a pairwise fashion using the Smith-Waterman algorithm. B. It doesn't take input from Fred. C. It is used for sequence assembly. D. It is a UNIX program. The correct answer is it doesn't take input from Fred. Which of the following is incorrect about Fred? A. It is a UNIX program. B. It doesn't give a probability score in output. C. It is used for base calling. D. It uses a Fourier analysis to resolve fluorescence traces and predict actual peak locations of bases. The correct answer is it doesn't give a probability score in output. Which of the following is incorrect about EULER? A. It is an assembly algorithm. B. It uses a EULER superpath approach, which is a polynomial algorithm. C. In this approach, a sequence fragment is broken down to tuples of 5 nucleotides. D. The tuples are distributed in a diagram with numerous nodes that are all interconnected. The correct answer is in this approach, a sequence fragment is broken down to tuples of 5 nucleotides. Which of the following is untrue about base calling and assembly programs? A. The first step toward genome assembly includes derive base calls. B. The first step toward genome assembly includes assigning associated quality scores. C. One of the steps is to assemble the sequence reads into contiguous sequences. D. There is no identifying overlap between sequence fragments. The correct answer is there is no identifying overlap between sequence fragments. The major challenges in genome assembly are sequence errors, contamination by bacterial vectors, and repetitive sequence regions. A. True. B. False. C. D. The correct answer is true. TIGR Assembler is a UNIX program from TIGR for assembly of large shotgun sequence fragments. A. True. B. False. C. D. The correct answer is true. VecScreen is a primarily aimed for sequence assembly. A. True. B. False. C. D. The correct answer is false.